Hi there. Okay, so we're now on to, we've covered diffusion as passive transport, osmosis as passive transport. We're now looking at the effect that osmosis can have on cells. Okay, so this is about water concentrations, it's about plant and animal cells. If they're exposed to different solutions, what can happen to them?
Okay, so in this we are going to be looking at how placing different cells, so either animal or plant cells, in different concentrations can change their appearance and how we would describe their appearance based on this. By the end of this, there's quite a long list of things you'll be able to do here because this is probably one of the hardest skills that comes up and one that always comes up in the exam. So you should be able to describe what happens to an animal or a plant cell when it's submerged in high water concentration. You should be able to state what a turgid cell looks like and describe how the cell will become turgid. You should be able to describe what happens to an animal and plant cell when submerged in a low water concentration, and state what a plasmolysed cell will look like and describe how a cell will become plasmolysed. So lots of words there for you.
Lots of new vocabulary and some quite lateral thinking skills involved. So a reminder, osmosis is a type of passive transport. That means it requires no energy, and it's a movement of water molecules from a higher water concentration to a lower water concentration through a selectively permeable membrane. It may sound like I'm repeating myself a lot here. I cannot stress this enough. Must have that definition absolutely memorized word for word.
Okay, so this is just a wee example again, just to remind your brain. So water concentration outside the cell is at 83%, water concentration inside the cell is at 64%. So based on our rules of osmosis that we know, water is going to move from the higher water concentration, which is outside, to the lower water concentration, which is inside. And if you think about that, if something is gaining water, it's also going to be becoming heavier because it's getting something else inside it. So it will be gaining mass. Is what we would say in biology. So it's gaining water, so it's gaining mass. If it was losing water, it would be losing mass, and it will be getting lighter in weight. So that's something to keep in mind whilst we go through that idea of mass.
It's key. This idea, water is heavy. Gaining or losing it, even at a cellular level, is noticeable on a scale. It's very heavy stuff. Okay, so what happens if you put a cell into really, really high water concentrations, such as the animal cells here that are in distilled water? The distilled is spelt wrong. It should be DIS, okay? But into distilled water, such pure water that these cells are in, what happens if the water keeps flowing in and keeps flowing in and keeps flowing in and keeps flowing in? Try and think of it like a balloon if you keep putting air in it and air in it and air in it and air in it. Well, this happens to animal cells, but different stuff happens to plant cells, which we're going to investigate over this wee PowerPoint.
Okay, so the effects of osmosis obviously depend on the concentration of the water in the solution. So obviously, if it's really, really high water, it's going to be really, really low salt, whereas if it's really, really high salt, it's going to be really low water. And you're always doing that thinking, where is the higher water concentration? Where is the lower water concentration? And again, this could very much be told to you in terms of salt or sugar. You have to do that conversion of, okay, this is 10% sugar, that means it's going to be 90% water because the rest is going to be water. So again, keeping that thought in your mind. And that's it. Summarised nicely there for you in case you have forgotten.
Okay, so cells can gain or lose water by osmosis, as we covered in the last one. Now, if the solution that cells are surrounded by changes the water concentration, this can have a big effect on cells. So say I was to put a cell into a high water concentration or a higher water concentration, then the cells will gain water because there's a higher concentration outside, lower concentration inside, water will flow in. Equally, the opposite is true. Low water concentrations will cause cells to lose water. If there's a high concentration inside the cell and a lower concentration outside the cell, water will flow out and it will lose. Now, if it's gaining water or if it's losing water, similar things happen to actual people. You know what happens to yourself. If you're very, very, very thirsty, things stop working. If you have drunk far, far, far too much water, you're not in a good place either. Okay, it's a similar situation for cells. Too much water, bad. Too little water, bad.
Okay, so in terms of osmosis in animal cells, there are certain things that can happen depending on if it's a higher water concentration or a lower water concentration it's in. So to start with, if an animal cell is placed in a higher water concentration than its contents, so say it is 100% water outside, whereas it's only 50% water inside, it will then gain water. So water is going to move from that higher concentration outside to that lower concentration inside. And just like a balloon, it could burst. It's going to swell first of all. So like if you get a swollen bit in your body, it starts to get fatter. That's what we mean by swelling. It's going to get fatter and fatter. And then eventually, if there's too much water in it, it's just going to burst. And you can see in this diagram, the cell membrane is actually the thing that bursts. It's no longer actually able to withstand the pressure. You can see it's broken up into little bits that are now almost inside the cell, and it's not quite got that neat outline it does above. So animal cells in a higher water concentration than inside the cell will burst.
Okay, this is something that's related to the PLP a little bit, but also it's why, say you've got somebody who's been trekking across a desert and they are dying of thirst, like literally dying of thirst, and you've got an IV, so one of those little bags of liquid that you could plug into their arm. You should never, ever, ever just put water into that IV to flow into their blood, because you put in pure water in, you will burst their red blood cells and you will harm that person a lot. It'd be bad. Instead, I don't know if any of you have ever been to hospital, instead you get saline solution, which is a very slightly salty solution, and that's at the same concentration as your own cells. So that helps prevent the bursting, as we can see here. Okay, so if an animal cell is placed in an equal concentration to its contents, it'll gain and lose water at the same rate. Again, water molecules don't sit still. It's not like you can say, you freeze. Well, you can with water, but not like inside a cell. Okay, so they don't stay put, but they'll cross the membrane, but then they'll come over in equal amounts. So as the cell does not gain or lose water overall, the cell will remain unchanged. Okay, and that's what a saline drip does. Is a saline drip, when they plug you in at the hospital, is all it's doing is putting a solution of basically slightly salty water into your bloodstream. Not really salt, but I don't know how else to explain it. But they put it into your arm, into your bloodstream. And the point is, it's an equal concentration to what your cells should be. And so they don't gain water, they don't lose water, they just stay exactly as they are.
Okay. The final thing that can happen is if an animal cell is put in a low water concentration. So the solution that is in has got a much lower water concentration than inside the cell. So maybe inside the cell is 60% water, outside the cell is maybe 30% water. So osmosis rules say that water is going to move from the higher to the lower concentration, so it's going to move outside the cell. And if water comes out of the cell, it's going to basically just shrivel up and get smaller and smaller and smaller, because you're taking all of the water out from inside it. It's being drained out of it, so it's going to shrivel up into a little ball. And it usually looks something like this in this diagram here at the bottom.
Now, the examples of the diagrams you have just seen are one type of diagram from one particular exam paper. They can appear in slightly different formats, but you're looking for a basic model of a cell, and then you're trying to examine, right, is any of them showing, I don't know, contents that have poofed to the outside of them, or is it looking smaller? Maybe the nucleus is looking bigger in comparison. So those are the kinds of clues that you're looking for. Very common exam question is, here's a cell, we've exposed it to 3 different solutions, which one was the higher water concentration solution that we exposed it to? So you're looking for the one that's burst.
And you can see the difference in cell membranes here. In that one where it burst, the cell membrane was no longer whole, whereas in this diagram, you can still very clearly see that solid black line, which is the cell membrane around the outside. That's the thing that's going to be bursting, so that's the thing that can kind of give you a clue.
Okay, so to summarize it, if it's in a lower water concentration than the original cell, it will lose water. Equal concentrations, it will stay the same. Higher water concentration than the original cell, water will flow in, it can swell and burst. So that's specifically to animal cells. Different things happen to plant cells because plant cells have a cell wall. Okay, so this happens to animal cells because they have no cell wall to help stop this from happening. So they can burst or they can shrink, and that's all because of lack of a cell wall. And that's an exam question. It came up in last year's exam, or not last year's, but the 2019 exam paper is basically a reason why an animal cell might burst or shrink, and that's because of a lack of a cell wall, which is why it's important that you know your cell structures and which types of cells you find them in.
Okay, so here is another wee practice question which we'll talk you through. So water concentration outside the cell, 83%. Water concentration inside the cell, 25%. So higher concentration outside the cell, lower concentration is inside the cell. So water is going to be moving from that higher region outside to that lower region inside the cell. As a result of that, it is going to start to swell, it's going to eventually burst, and in this case it's also going to be gaining mass. So water is going to move in through osmosis, the cell is going to start to swell, and then it's eventually going to have it's going to burst and that cell membrane will no longer look the same.
Okay, so it's key to have that language point in there. The cell is going to gain water, the cell will burst because water is moving from the higher concentration outside the cell to the lower concentration inside the cell, and it is an animal cell, so it can burst.
Okay, so osmosis in plant cells, similar idea, different words, and the reason for this is they have that cell wall, and that cell wall is crucial because it prevents the cell from bursting. So we're going to start by looking at what happens if you put a plant cell in higher water concentration than its content. So again, maybe S is going into an 80% solution where maybe there's only 20% in the cell. There's a higher concentration in the solution that it is in. So in this case, water is going to be moving from higher to lower concentration. It's going to move inside the cell. In this case, it will still swell, but it won't burst. And that is because of that cell wall, that cellulose cell wall in our plant cells. It means that it can't burst. It's keeping that structure and that shape to it. So instead, what we say is it becomes turgid. So turgid is basically, it's when it's swollen up, but it's a plant cell. The word we use is turgid.
Okay, now the vacuole you also notice from the middle is also large as well, so that's another feature of a turgid cell. So again, if a plant cell is placed in an equal water concentration to its content, so the same outside as the actual plant cell on the inside, it'll gain lose water at the same rate, and because it doesn't gain or lose water overall, the cell will look the same.
Okay, finally, if it's put in a lower water concentration than its contents, the cell is going to lose water. So say it's in a 20% solution outside, inside the cell is 50%, water is going to be moving out of the cell by osmosis. So in this case, it's going to be moving out of the cell to the outside environment. And in this case, we would say it becomes plasmalized. So plasmalized is that definition for when water's come out of the cell. If you look at this diagram here, you will see the vacuole is much, much smaller. You can also see that the cytoplasm is coming away from the cell membrane as well. It's looking almost like someone's trying to suck out the inside and it's slowly being peeled away from the edge. This is a plasmalized cell. So this is because all the water, or not all the water, but some of the water has been moving out of the cell until it's at equilibrium.
Yep, so two features of the plasmalized cell, small vacuole, and the membrane has pulled away from the cell wall. Basically, this is empty space in the gaps that you can see. Essentially, there's the cell wall stays where it is because it's a nice firm structure, but there's nothing really there in the space between the cell wall and the membrane. Okay, now that's not an ideal situation. Plants can't live long at that. A plant that's got mostly plasmalized leaf cells, you'll notice that the leaves are drooping because they can't hold themselves up very well. So the leaves will be drooping, maybe quite saggy, maybe feeling a little bit fluffy is the word I want to use for those. But the idea is that it's not going to last very long in that situation, but it'll keep draining the vacuole, using up that spare store of water until it has nothing left, and then it will start to die. So to summarize, when the plant cells are placed in solution, in a lower water concentration outside the cell, so the cells will lose water and become plasmalized, so that feature of tiny vacuole, maybe you might not even see the vacuole because it's so small, and membrane pulled away from the wall. Equal water concentration, the cells will remain the same, and a higher water concentration outside, water will flow in, causing the cell to swell up, the vacuole gets really big, the cell wall might push out slightly, and that's where it's called turgid. So those two words, turgid, plasmalized, very, very important.
Okay, so again, this is another practice question. You can try it yourself, but we'll talk through it anyway. If you have a plant cell, inside the cell is 76% water concentration, outside the cell is 25% water concentration. So inside our cell, higher water concentration, outside our cell, lower water concentration. Water is going to move from higher to lower, so water is going to move out the cell. So in this case, water is moving out the cell is going to become plasmalized as it will lose water when the water is moving from that higher concentration on the inside to the lower concentration in the outside, which is summarized there for you and where you would get marks.
Okay, so keywords that we need to know for this. And it's mainly this idea that you need to know what conditions would cause these words on the left to happen. So a burst cell would be an animal cell that's been exposed to a higher water concentration solution than its contents. Water's flowed in, and then it's burst.
Okay. So a shrunken cell is an animal cell again that's been exposed to lower water concentration. So the water has then moved out of it, so it is starting to shrink and become smaller and smaller, and it is losing water.
a turgid cell is going to be a plant cell that has been exposed to a higher water concentration than its contents. Water's flowed in, vacuole big, cell wall slightly bowing slightly just to show the pressure of it, but again not burst because of that cellulose cell wall has saved it from bursting.
And finally, plasmolyzed, again, it is a plant cell. It is one that is in a lower water concentration, so water is going to be moving out from it, and it's got that look of really small vacuole. You get your cell membrane, your cytoplasm looking like they've been pulled away from the side, and that is because it is losing water to the external environment.
Okay, so you have to be able to, and this is the key skill, describe what happens to animal and plant cells when they're submerged in higher water concentration solutions. So slightly different animal compared to plant. State what a turgid cell looks like. So that is just the plant cell with the big vacuole and it's looking slightly swollen, and it's because it's been in a higher water concentration. Describe what happens to animal and plant cells when submerged in a low water concentration. So again, slightly different things happening there. And then state what a plasmolyzed cell looks like. So that's tiny vacuole, membrane pulled away from the cell wall, and that's because it's been submerged in a lower water concentration, causing the cell to lose its water. Now, there are loads of experimental type questions in the past papers that refer to this. They sometimes use eggs as model cells, so they're pretending that the egg is the cell and watching it gain and lose water. There are some about weighing potatoes. So the idea is they fling shards of potatoes into different salt solutions and see if they gain or lose mass, i.e. gain or lose water. These are so worth practicing.
Something that does sometimes come up, and it's came up with the potato one especially, is they sometimes talk about the idea that you have to dry the potato before weighing the mass of it, and that is just the idea that if something gets wet, there's going to be excess water on the outside of it. Like if you go out in the rain, you're going to have water down your skin. That's not part of the water in you, that's just excess water. You would towel dry yourself. It's the same thing. If you're going to weigh something to find out its water mass, you need to dry it first to get all the water off the outside of that, and that is sometimes something that comes up. It's usually only worth 1 mark, but it's really useful to know that the drying it to get its dry mass is really important.
So highly, highly recommend. If you feel quite confident with this, get to the SQA website, get onto their past papers and try and find those questions. I think they're egg-based, potato-based, and there's another one about beetroot or something like that.
It's quite often the second question in sections 2 of pretty much every year's exam that you're going to do.
Practice those questions, see how you can link the changing numbers of the massive stuff to this idea of spotting the higher concentration and lower concentration. You will be fine. If you can spot the higher water concentration, the lower water concentration, you should be fine. That's the whole basis behind this, is being able to spot the water concentration, and then you'll be good.
Yep.
Okay, so next section is, I think, the final section of this key area, which is active transport.